Hello everyone, my name is Claire and this is Biology in Minutes. Today, we will cover 5 steps to get you started in the oldest DNA sequencing technique, the Sanger DNA sequencing by capillary electrophoresis. Now you might be wondering, what is DNA sequencing? DNA sequencing is finding the precise order of the 4 components of a DNA sequence in a DNA fragment. These 4 components are called nucleotides, and they are linked to each other like train wagons in what we call a DNA strand. These nucleotides also match in 2 pairs. So you have those 2 pairs, the first one and the second one. In a DNA sequence, the nucleotides in the first strand are mirrored in the second DNA strand with their pair nucleotide. So why do scientists want to sequence DNA? Because when we sequence, we can identify changes or mutations in a sequence. We study these mutations because they can have an impact. For example, one mutation can give a pathogen the ability to resist to a treatment. Or, in the case of SARS-CoV-2, the virus that is at the origin of COVID-19, new variants. How do I sequence this DNA fragment using the Sanger technique? Here are 5 simple steps, starting with the DNA fragment. Step number 1 is very simple. We heat up the DNA to separate the 2 strands. We want to separate these strands so that we can attach a short sequence called a primer to the first strand. Step number 2, we mix the following reagents in a tube. The DNA strand you want to sequence, a starter, what we call a primer. It's a short sequence that we designed to mirror the beginning of the fragment. Nucleotides, these 4 components of DNA that we discussed before, and some special nucleotides. So what makes them special? They present 2 differences compared to the regular ones. First, they are unable to connect to another nucleotide on one side. And second, they are of different colors. So we get 4 special nucleotides of 4 colors. We also add an enzyme, which is called DNA polymerase, and this enzyme is a tool that links nucleotides one to another. Step number 3, let it run. The enzyme will do its job and recreate the first strand by taking nucleotides one by one from the mix, and add these nucleotides one after each other in the complementary strand, starting with our primer. But you remember these special nucleotides that we added in step number 2? They can link on one side, but cannot on the other one. So if the enzyme uses one of these special nucleotides, the reaction stops. And because this reaction is happening hundreds and hundreds of times, the new strand will grow hundreds of times and randomly contain a special nucleotide. So, at the end of the reaction, we obtain a lot of DNA fragments, all of different sizes, and finishing by a different color, which leads up to step number 4, the puzzle. The newly synthesized DNA fragments are sent into a small pipe with an electric current. The fastest to reach the end are the shortest. This way, we can sort the fragment by their lengths, depending on which one arrives first. And because all of them are from a different color, with a special laser and camera, we can capture their color, and from this color, reconstruct the sequence. For example, if the first fragment to reach the camera finishes by an orange nucleotide, the first nucleotide is orange. And then if the second fragment finishes by a blue nucleotide, the second base is blue. And if the third one finishes by a red nucleotide, the third one is red, and so on, and so on. Step number 5. We are finally getting what we wanted from the start, which is the sequence. But because what we obtain is the matched strand from the original one, we need to revert this sequence. Here you go. After this, we analyze the sequence by comparing it to the sequence that we know. How do we do that? I'll show you in the next video.